Namaste students, I am Javin Dr. Aziz Mohammed Khan from Department of Food and Biotechnology, Faculty of Agriculture and Marine Science, Jyoti Vidyapith University, Jaipur. Today I am going to discuss about the topic inhibition of starter culture by chemicals, detergents and some miscellaneous inhibitors. So there are some various kind of factors which will inhibit the growth of microorganisms which are involved in the fermentation process. They are not necessary, they are the outsider factors which are mostly uh, involved in the fermentation. They may be the raw material. These inhibitors may be found in the raw, may be present in the raw material, or they can be found in the equipments. So these inhibitors will inhibit the growth of microbes which are present in the fermentation process. They are actually not required. So in this video, I am going to discuss about the inhibitors of starter culture from chemicals and detergents. First, uh, chemicals and detergents which are used for the cleaning process. Okay, uh, they are used in the cleaning process of the dairy plant. Means they are used in the cleaning of the fermenters. Okay, suppose they are using the for cleaning of the fermentation, sanitization of the fermentation. Okay, but if they are remain in the fermenter, then their presence can disturb the fermentation process. Okay, how they will disturb? They will disturb the fermentation process because they are in the dairy plant, and when they are present in the dairy plant, they can inhibit the growth of microbes because they are detergents, they are disinfectants. Okay, they can inhibit the growth of microbes which are involved in the fermentation. Okay, they can inhibit the growth of cultures. Okay, the residues of these compounds include alkaline detergents, chlorine-based materials, hydrophores, quaternary ammonium compounds, and some ampholites. Okay, these are some undesired compounds, but these detergents, these compounds can inhibit the growth of uh, culture, microbial culture. Okay, yogurt culture are more tolerant to the activities of these residues at the inhibitory levels. Contamination of starter mix with these compounds is majorly due to human error. They are actually these detergents, disinfectants are found in the process due to the some human errors. They are actually not found, but they are found because of some human errors. Some undesired, some unauthentic functions will cause the presence of these detergents and infections, disinfectants. And when they are present in the fermenters, they are present in the fermentation process, they will inhibit the growth of culture, definitely, because they are detergents, they are disinfectants. So definitely they will inhibit the growth of cultures. Now we are going to discuss about some miscellaneous inhibitor. What is the meaning of the miscellaneous inhibitor? Means they are not added to the fermentation mixture, but they are by default. They are in the fermentation process by usual process. They are the usually raw material presents which can inhibit the growth of microbes also. Some antibodies, some antibodies such as lactase and agglutinins, which are present in the milk. Okay, these antibodies are present in the milk also, but these antibodies can also inhibit the growth of starter cultures. Okay, they can also these antibodies can also inhibit the microbes which are involved in the fermentation process, and what how they can be found in the milk because they are heat sensitive. They cannot be they can be found in the milk if the process is not heated. If the milk is not heated, okay, so heat the milk must be heated so that these antibodies will not be found in the dairy process, because if the milk is not processed, if the milk is not heated, then the antibodies which are natural antibodies, they can inhibit the growth of some microbes which are involved in the fermentation process. Simple. Leukocytes in the mastitis milk can cause phagocytosis of the starter cultures of fermentation. Thiocyanate, which is present in the late lactation milk, may also inhibit the growth of fermentation process microbes. And heating of starter results in no significant improvement of the end product. We can attribute some other inhibitors of the environment pollution, such as insecticides. Okay, these insecticides can inhibit the microbial growth. Volatile and non-volatile compounds can also inhibit the growth of microbes. Such volatile compounds may include fatty acid, formic acid, formaldehyde, acetonitrile, chloroform, and ether. Okay, these chemical agents which are present in the normal process or normal environmental pollution vector can inhibit the growth of fermented nutrition microbes. The microbes which are involved in the fermentation process, but they will not inhibit the growth of microbes at lower concentration. Okay, if they are in lower quantity, if these compounds, fatty acid, formaldehyde, acetonitrile, chloroform, uh, insecticides, okay, if they are in low quantity, they will not inhibit the growth of microbes. But when they are higher in their concentration, they will inhibit the growth of some microbes, including the streptococcus and lactobacillus pneumoniae. So that's all about the uh, inhibition of microbes by some factors. Our lecture is over here. Thank you very much.